Hi everyone, so today we'll be talking about immunology. So immunology is one of the difficult topics in the Part 1 Irish Dental Council exam. We'll try to cover the most important topics and the basics for you to get a better understanding. So the number of immunology questions has been increasing every year. The highly repeated questions are antibody structure and function, primary versus secondary lymphoid organs, the main antibody type in saliva, type of phagocytic cells, MHC class 1 and class 2, T-cell receptor complex, complement pathway and activation, some vaccination terms, monoclonal versus polyclonal antibodies, innate immunity components, and hypersensitivity reactions. So we have many barriers preventing the pathogens from entering our body. So we have the skin and mucous membrane, we have tears and saliva that contain lysosomes and antibodies, we have the stomach acid as well, and our normal flora. So our immune system is made up of organs like the bone marrow and thymus, cells like lymphocytes and monocytes, and molecules like antibodies and cytokines. So all cells of the immune system originate from the bone marrow. So we have myeloid lineage, which generates the granulocytes and monocytes, and we have the lymphoid lineage, which generates the T cells, B cells, and natural killer cells. Primary lymphoid organs are where the immune cells form and mature. So for the B cells, they form and mature in the bone marrow. The T cells form in the bone marrow and mature in the thymus. And the remaining immune cells all originate from the bone marrow. The secondary lymphoid organs are where the immune cells reside and where they encounter pathogens and launch a response. The lymph node and spleen are examples of secondary lymphoid organs. So the lymph is actually the medium by which the lymphocytes and pathogens are taken to these secondary lymphoid organs. So we have two divisions of the immune system. We have the innate immunity. So it provides rapid, non-specific defense against pathogens, and it's the first line of defense and acts immediately upon encountering the pathogen. So it's non-specific and it has no memory. So the components of the innate immune system are general immune cells like neutrophils, basophils, macrophages, dendritic cell, natural killer cells, as well as soluble molecules like complement proteins and cytokines. On the other hand, we have the adaptive immunity. So the cells of the adaptive immunity are the B cells and T cells, and they generate a specific defense against the pathogens. And so their response is slower, and they also develop memory cells, which can later provide a better and stronger response following a subsequent exposure. So the leukocytes are divided into granulocytes and agranulocytes. Granulocytes are white blood cells that contain granules in their cytoplasm and have an important role in the immune response. The neutrophils are the most abundant white blood cell, and they are crucial in controlling bacterial infections. They are also phagocytes. Eosinophils play an important role in allergic reaction and parasitic infection. We also have basophils and mast cells. So they're called basophils when they circulate in the blood, and they're called mast cells when they enter tissue. They play a crucial role in allergic reaction because they release histamine and other inflammatory mediators, and they contain receptors for IgE antibodies. Lymphocytes and monocytes are considered agranulocytes because they lack cytoplasmic granules. We'll talk about lymphocytes later on during this lecture. Monocytes and macrophages are similar to basophils and mast cells. So they're called monocytes when they're in the blood, and then they're called macrophages when they enter the tissue. So the function of macrophages is phagocytosis, secreting cytokines, and antigen presentation. We have dendritic cells, which are the most efficient antigen-presenting cells. Antigen presentation is crucial for the function of T cells because they're unable to process or detect pathogens without having the antigens processed and presented to them. We also have natural killer cells, which kill virus-infected cells and cancer cells. The natural cell can eliminate pathogens by releasing two types of enzymes. So we have perforin, which causes pores in the pathogen's membrane, and it also releases gran enzymes, which causes lysis and apoptosis of the cell. Another function of natural killer cells is cytokine release, so it's able to release interferon gamma and tumor necrosis factor and others, which are pro-inflammatory. So when a cell becomes infected, it actually doesn't express as much MHC molecules, so that makes it detectable by the natural killer cell. This is called missing self. The natural killer cell is able to detect antibody-coated virus-infected cells or tumor cells. So we'll discuss the structure of antibodies later on, but the antibody has a portion called an Fc portion, and the natural killer cell has an Fc receptor. So the Fc receptors of the natural killer cell will bind to the antibody, and this will trigger the release of their enzymes, and this is called antibody-dependent cellular cytotoxicity. We have three types of phagocytes. So we have macrophages, neutrophils, and dendritic cells. So phagocytosis is the process of engulfing the pathogen and digesting it and eliminating its waste. So the phagocyte is first recruited to the site of infection by a process called chemotaxis, which means it becomes attracted by chemicals released. The pathogen is then engulfed by the phagocyte and it's packaged into a phagosome. The phagosome fuses with a lysosome, forming a phagolysosome, which then destroys the pathogen and then the waste is eliminated. So what actually is an antigen? An antigen is any substance, like a protein or a lipid, that the immune cell detects as foreign and it triggers an immune response. An epitome is a specific region of an antigen, so one antigen can have many different epitomes. Antigen presentation is the process of cleaving antigens into smaller peptides. These peptides are then associated and attached to self-surface proteins, and they are presented on the surface of the antigen-presenting cell. So we have three types of antigen-presenting cells: dendritic cell, which is the most efficient one, the macrophages, and B cells. So just to remind you, both macrophages and dendritic cells are phagocytes and antigen-presenting cells. So how do innate immune cells like macrophages and neutrophils detect pathogens? They actually have receptors called pattern recognition receptors. The main one is called toll-like receptor. These receptors are able to detect pathogen-associated molecular patterns, which are molecules found on the surface of pathogens like peptidoglycans and polysaccharides. The complement system plays an important role in the innate immune response. It consists of proteins that are present in the plasma and they're produced in the liver. They are present in an active state, and once activated, they initiate a cascade, leading to a central event, which is the C3 cleavage into C3a and C3b proteins by a C3 convertase enzyme. So the complement cascade is activated by different methods. So the classical pathway, as its name suggests, is activated by antigen-antibody complexes. So when there is an infection, the antigen antibodies bind, activating the C1 protein, which eventually leads to the activation of the C3 convertase enzyme. Another method by which complement proteins become activated is when a mannose-binding lectin, which is a protein produced in the liver, identifies sugars present on bacteria and viruses. This is called the lectin pathway. The alternative pathway is when the C3 undergoes spontaneous hydrolysis. So the C3 is usually present in an active form in plasma. It becomes activated when it encounters the surface of a pathogen, so it doesn't wait for a signal or an event like antibody binding. So the C3A generated will induce inflammation. The C3B will label the pathogens for phagocytosis. C5A also induces inflammation and is a chemoattractant. C5b, C6, C7, C8, and C9 actually form a pore in the pathogen, which is called a membrane attack complex and causes death. So that summarizes the function of the complement system. So it's opsonization, inflammation induction, membrane attack complex formation, chemotaxis, and immune complex clearance. Cytokines are small proteins used in cell-to-cell signaling in the immune system. They are produced by various cells like the macrophages, lymphocytes, mast cells, as well as endothelial cells and fibroblasts. So we have different families of cytokines. We have the interleukins, which coordinate immune responses. So we have the interleukin-1, which is pro-inflammatory and results in a fever. We have the interleukin-2, which stimulates the growth and differentiation of T cells. We have interleukin-3, which stimulates the bone marrow, interleukin-4 and 5, which stimulate antibody production, and we have many others. We have the interferon family, which have strong antiviral activity, and the tumor necrosis factors, which also mediate acute inflammation and activate other immune cells. If you're unable to memorize the specific function of these cytokines, then you can always try to list a broad function like activation of immune cells, regulation of immune cell development, or anything within these lines. So now we'll move on to the adaptive immunity. So we have the humoral response and the cell-mediated response. Humoral immunity uses antibodies to target extracellular pathogens. So B lymphocytes develop and mature in the bone marrow. They then reside in the secondary lymph nodes. A naive B cell is one that hasn't been exposed to an antigen yet. After it becomes exposed, it becomes activated and differentiates into plasma cells, which produce antibodies. An antibody has 4 chains. It has 2 identical heavy chains and 2 identical light chains. Each chain has a variable portion at the tip and a constant portion closer to the base. The arms at the top is where the antigen binds. This region is also called the FAB region. This is different between each antibody. The base of the antibody is called the constant portion. It's also called the FC portion, which stands for fragment crystallizable. So this region is the same in different classes of antibodies. So it's the same in all IgG antibodies. It's the same in all IgA antibodies. So this region determines the class of antibodies and therefore determines the effector functions of the antibodies. So we have 5 classes of antibodies. The most abundant one is the IgG, has many functions like opsonization, neutralization, and complement activation. We have the IgM, which is the first antibody produced in the immune response. After that, the IgM undergoes class switching, which means a different isotope of antibodies produced with the same antigen specificity. We have the IgE antibody, which is present in secretions like tears, milk, and saliva and provides mucosal immunity. We have the IgE antibodies, which are important in allergic reactions and parasitic infection. And we have the IgD, which acts as a B-cell receptor. We'll talk about B-cell receptors later in this lecture. So antibodies have many functions. So we have neutralization, which means the pathogen is blocked and is made inactive. We have opsonization, which is labeling the pathogen for phagocytosis, complement activation, agglutination and precipitation, which means clumping the pathogens together. And we have the antibody-dependent cytotoxicity. So the antibody coats the pathogen, allowing the natural killer cell to detect and kill it. The immune cell recruitment, which overlaps with opsonization and complement activation. A B-cell receptor is present on naive B cells, and it's essentially an IgM or IgD antibody and a signaling protein. This signaling protein is called the Ig alpha or Ig beta, which signals into the B cell when an antigen binds, and this releases the bound antibody. So basically, the antibody that was bound to the membrane is no longer a receptor, it now acts as a soluble antibody. This happens after the B cell differentiates into a plasma cell upon activation. So we'll go over the steps of a B cell activation. So the antigen is recognized by a macrophage, a dendritic cell, and presented to the B cell, or the B cell detects it directly. The B cell becomes activated with the help of a T cell. This is called a T cell dependent response. So when the T cell releases cytokines and stimulates the B cell, it produces a better response. After activation, the cells will undergo proliferation, they will differentiate into plasma cells, they will undergo class switching and somatic hypermutation, which we'll discuss in the next slides. Monoclonal antibodies are antibodies that are a single B-cell clone that target a specific epitope on an antigen. They are used in the treatment of specific cancers and other types of immune therapy. On the other hand, polyclonal antibodies are multiple B-cell clones that recognize multiple epitopes for broader antigen binding. An example of their use is antivenom used for snake bites. So when a B-cell becomes activated by the presence of an antigen, they differentiate into plasma cells, and some cells differentiate into memory B cells, which provide immunological memory and a better response upon subsequent exposure of the same antigen. So we'll go over the concept of class switching. So once a B cell receptor, which is essentially an IgM antibody bound to the B cell membrane, binds to an antigen, it becomes activated and this IgM antibody will be released into the serum. So that is why the IgM antibody is the first antibody produced during an immune response. After that, a specific enzyme alters the constant heavy chain region in the structure of the antibody, and so the antibody will undergo class switching. So it will form a different class like an IgG or an IgA or an IgE depending on the cytokines and the signals. Our body's ability to generate diverse antibodies is very important to be able to recognize and attack different pathogens. The generation of diverse antibodies occur during 2 main phases. So initially it happens in the bone marrow, where different gene segments that are called the variable, diversity, and joining genes undergo random recombination. Antibody diversity is also generated post-antigen activation when specific enzymes mutate the variable regions on the antibody. This is called somatic hypermutation. The other division of the adaptive immunity is the cell-mediated immunity. These cells are CD4 positive T lymphocytes. They subdivide into T helper 1 cells, T helper 2 cells, T helper 17 cells, and T regulatory cells. So the T helper cells don't actually attack or kill the pathogens directly. They promote and activate other immune cells and other parts of the immune system. The CD8 positive T lymphocytes, also called the cytotoxic T cells, are able to directly attack and kill the pathogens by releasing perforin and gran enzymes, and thus killing the cells by inducing apoptosis. So if you remember, the target of the humoral immunity are extracellular pathogens, And the target of cytotoxic T cells are intracellular pathogens and cancer cells. So what is the major histocompatibility complex? So they're a group of genes that encode cell surface proteins. Their main job is to present peptide antigens to T cells and help the immune system recognize self from non-self. Class 1 MHC are present on all nucleated cells, and they present endogenous antigens to CD8 positive T cells. So what does that mean? So if you remember, the target of cytotoxic T cells are intracellular pathogens. So intracellular pathogens like viruses replicate inside the host cell. So the infected host cell will have endogenous antigens. These antigens will combine with the MHC class 1 molecules, which will be carried to the surface of the antigen-presenting cell and will be presented to the cytotoxic T cell. The class 2 MHC molecules are present mainly on antigen-presenting cells. So what does that mean? So when an antigen-presenting cell encounters an extracellular pathogen or microbe, it processes the antigen. It binds to the MHC class 2 molecule and is carried to the surface to be presented to the helper T cell. So the MHC bound to the antigen presented to the T cell will actually bind to the T cell receptor. The T cell receptor complex is made up of two protein chains, an alpha and a beta chain, that recognizes and binds the antigen, a CD4 or CD8 marker, which acts as a co-receptor for the MHC molecules, and a CD3 protein, which is responsible for signal transduction and transmission. So both CD4 and CD8 T lymphocytes require antigen presentation and stimulation. Similar to antibodies, the body also generates diverse T cell receptors to be able to identify different pathogens. So they undergo diversification in the same manner by gene segment recombination. So we'll just go over the steps of T cell development and activation and function. So the T cells develop in the bone marrow, they mature in the thymus, in the secondary lymph nodes like the spleen or lymph nodes. They will have the antigens presented to them by the dendritic cells or other antigen-presenting cells. The antigens will be processed and attached to the MHC molecules, which will be carried to the surface and binds to the T cell receptor. Then a series of signals will induce the proliferation and differentiation of T cells, which can then perform their function. So we'll just go over the immune response together. So the pathogen will first encounter the varied defenses of the body. So we have the physical barriers, the chemical barriers, and the biological barriers. The cells of the innate immunity then come into action. So they perform their specific functions like phagocytosis, recruiting other cells, releasing cytokines. The complement system also becomes activated, leading to the formation of a membrane attack complex, as well as the other functions like opsonization and inducing inflammation. So the antigen-presenting cells need to present the antigens to the T cells. Endogenous antigens are combined with MHC class 1 and they're presented to the cytotoxic T cell. Exogenous antigens are internalized and processed by the antigen-presenting cell and they're combined with the MHC class 2 molecules to be presented to CD4 positive helper T cells. So the peptide or antigen plus the MHC molecule actually binds to the T cell receptor of this T lymphocyte. And then a series of internal signals will activate the CD4 helper T cells to differentiate into different subtypes. So we have the T helper 1, T helper 2, T helper 17. So their main function is to produce cytokines and also activate other immune cells. Once a cytotoxic T cell is activated in the same manner, it kills the cell directly by releasing perforin, which forms pores in the pathogen, as well as inducing apoptosis by binding to the pathogen and activating death receptors. So the B cells are activated when an antigen binds to the B cell receptor. At the same time, CD4 cells become activated and they also release cytokines that enhance the B cell response. The B cell will proliferate and differentiate into plasma cells, which will then release the IgM antibody bound to the receptor. This IgM antibody will perform its function and then it will undergo class switching. And of course, some of the generated T and B cells will form memory cells that will become active upon a subsequent exposure. Hypersensitivity reactions are exaggerated, inappropriate immune responses that actually cause tissue damage rather than protection. So there are 4 types. 3 of them are antibody-mediated and the type 4 is T-cell mediated. The type 1, where the immediate reaction induces anaphylaxis. So upon first-time exposure to the allergen, the body produces IgE antibodies and they bind to mast cells. Upon re-exposure, the allergen cross-links the IgE that is bound to the mast cells, and this causes rapid degranulation, releasing histamine, leukotrienes, and prostaglandins. These cause vasodilation, bronchospasm, and other effects seen in anaphylactic reactions. The cytokine interleukin-4 plays an important role in this reaction because it induces class switching from IgM to IgE. We have the type 2 hypersensitivity, which is mediated by IgG or IgM antibodies. An example of this hypersensitivity reaction is hemolytic anemia. So the body will produce antibodies directed against cell surface antigens, and this will lead to cell destruction by antibody-dependent cellular toxicity or membrane attack complex formation. Type 3 hypersensitivity occurs when antigen-antibody complexes form in the circulation and they start to deposit in tissue and in vessels. When this happens in excess, the complement system becomes activated and induces inflammation. So this happens in diseases like systemic lupus or rheumatoid arthritis. We also have the type 4 hypersensitivity, which is a cell-mediated immune response. This results in a delayed type of response. So an example of this is contact dermatitis or the tuberculosis skin test. In this reaction, T cells become active and they induce inflammation and cause direct cell cytotoxicity. Macrophages play an important role in this reaction. So we have different types of vaccines. We have the live attenuated, kill, toxoid, and subunit. So they've asked about the definition of an attenuated vaccine before. So an attenuated vaccine is one that contains a live pathogen, but it's been weakened. So the pathogen is live and can induce an immune response. However, it's weakened, so it won't harm the host. In a vaccine, we have the active ingredient, which is the virus or bacteria. We have an adjuvant, which is basically a chemical added to boost the immune response to the vaccine. We also have antibiotics, stabilizers, and preservatives. So we have two different types of immunization. So we have active immunization. So for instance, when you actually contract the disease or when you get vaccinated for it, it's called active because the body actively produces antibodies. So it gives long-term protection. We also have passive immunity. So that happens when we receive already made antibodies. So for instance, the baby receives antibodies from the mom's breast milk. So B cells can be used in immune therapy. So monoclonal antibodies can be used in the treatment of cancer or autoimmune diseases. T cells can also be engineered and used in the treatment of cancer. So we have a treatment called CAR T cell therapy. So in this therapy, the T cells are extracted from the patient and a gene encoding a chimeric antigen receptor is encoded, usually by a viral vector. The receptor will then be able to bind to a specific antigen on a cancer cell. The T cell is then reinfused back into the patient's body. This treatment can be used in the treatment of blood cancers like leukemia or lymphoma. So the topic of immune therapy is quite advanced and very difficult to cover all of it. If you have the time, you can definitely go through research articles or extra reading material.